Hello, everyone. I'm Xiaoyun Li from Pathology Department, Santou University Medical College. Welcome to this online course of inflammation. What is inflammation? Let me give you an example. Look at this picture. The affected skin is red, swollen, and has multiple blisters. It is warm and painful to touch. Microscopically, these blisters are full of a fluid called serous exudate. Actually, the production of this lesion is due to an inflammatory reaction caused by herpes zoster virus. We'll go through the contents of inflammation through 8 sessions. Today's topic is definition and basic pathological changes of inflammation. After this session, you should be able to define inflammation, list the causes of inflammation, describe the basic pathological changes of inflammation, and the beneficial effects and potential harm of exudates. So, what is inflammation? It is a defensive response of living organisms with a vascular system to all forms of injuries. Tissue injuries trigger the body's inflammatory reaction. But this cannot happen in dead organisms and those without vascular systems. Why? Because vascular reaction is the indispensable component of the inflammatory process. Tissue injuries rapidly cause vascular reaction. This response involves changes in vascular caliber, increased blood flow, and vascular permeability, which will result in exudation of fluid, plasma proteins, and leukocytes. The inflammatory exudate can contain and eliminate the cause of injury, remove necrotic cells and tissues, and initiate the process of repair. Therefore, inflammation is a response involving blood vessels, host cells, proteins, and other mediators. And it is also an integrated process comprising tissue injury, protection against injury, and repair. The defensive effects of inflammation are realized by exudation. Inflammation is a protective response of the body, but it can also cause considerable harm to the body. The components of inflammation that destroy and eliminate pathogens and dead tissues can also injure normal tissues. If the reaction is very strong, prolonged, or inappropriate, tissue damage may even become the dominant feature. Massive exudation in vital organs may result in severe consequences. Repair may cause tissue fibrosis and adhesion because repair often involves the proliferation of granulation tissue. Persistent inflammatory stimulation could induce tumors. So, everything has two sides. Now, let's look at the causes of inflammation. Inflammatory reactions may be triggered by a variety of stimuli, including physical, chemical, and microbiological agents, tissue necrosis, foreign bodies, and immune reactions. Not all inflammatory reactions are induced by infections. However, microbial pathogen is the most common cause of inflammation. The basic pathological changes shared by different types of inflammation include alteration, exudation, and proliferation. I have mentioned that inflammation is an integrated process involving tissue injury, protection against injury, and repair. Here, alteration is the injury. Exudation refers to the protection against injury, and proliferation represents process of repair. For acute or early stage inflammation, alteration or exudation predominates, while in chronic or late stage inflammation, proliferation prevails. Alteration refers to degeneration and necrosis of tissues. We have discussed degeneration and necrosis in chapter of tissue injury. Alteration may occur in both parenchymal and mesenchymal tissues. Causes of alteration include the initial inflammatory stimulus, the secondary local circulatory disturbances, and the effects of inflammatory cells and mediators. As already mentioned, tissue injuries rapidly cause vascular reaction, which is followed by inflammatory exudation. What is exudation? Exudation is the movement of fluid, plasma proteins, and leukocytes from blood vessels into extravascular tissue at the site of injury. Exudation is an active process which is associated with increased vascular permeability. The goal of exudation is to bring leukocytes and proteins to the inflamed area to clear noxious stimuli and damaged tissue, exerting their protective effects. Exudation represents the body's defense against injurious stimuli and is the most important feature in inflammatory reaction. Extravasation of protein-rich fluid and leukocytes leads to the formation of exudate. Exudate builds up in the interstitial spaces, resulting in edema, which, if accumulate in body cavity, will produce inflammatory hydrops. In this picture, we can see the widened interendothelial spaces, indicating increased vascular permeability, which contributes to the exudation of water, fibrin, complements, antibodies, and leukocytes. Here we can see the neutrophils are polymorph nuclei with their bi- and tri-lobed nuclei. These are macrophages. The nucleus is kidney-shaped and lightly stained with a clear nuclear membrane. In this area, we have fibrin, which appears as red, thread-like, cross-linked substance. These are lymphocytes with big and darkly stained nuclei. Both transudate and exudate can result in edema or hydrops. So how do we differentiate them? First, let me briefly introduce the formation of transudate and exudate. Intravascular hydrostatic pressure of a capillary bed promotes outflow of water from the blood vessels into tissues, whereas intravascular colloid osmotic pressure prevents this process. Normally, these two pressures are in a balanced state. When intravascular hydrostatic pressure increases or colloid osmotic pressure decreases, this balance is disrupted, resulting in ultrafiltration of blood plasma. This causes the outflow of water through blood vessel and the formation of protein-poor transudate. In other words, transudate is the result of plasma ultrafiltration. During inflammation, increased interendothelial spaces allow protein-rich fluid to easily leak out into the extravascular tissues through the vessel wall, forming protein-rich exudate. So, exudate is the result of increased vascular permeability. Now, we can easily understand the difference between exudate and transudate. Look at this picture. The transudate is protein-poor and clear, has few cells, and low specific gravity, and doesn't coagulate. In contrast, the exudate is protein-rich and cloudy, has some cells and higher specific gravity, and it coagulates easily. What are the beneficial effects of exudate? Exudate dilutes and neutralizes toxins, delivers nutrients and oxygen to infiltrating leukocytes, and removes metabolic waste. Antibodies and complements can contribute to microbial clearance. The fibrin meshwork limits the spread of pathogens, provides a surface to facilitate leukocytic phagocytosis of pathogens, and also becomes the scaffold for tissue repair. The leukocytes can kill the microbes and eliminate necrotic tissues. Pathogens and toxins in the exudate stimulate host immune response. However, excessive exudation may be harmful. It could cause compression and obstruction, organization and adhesion. For instance, exudate in the pericardial cavity may restrict cardiac activities. Severe edema of larynx may cause asphyxia. Massive fibrin in the exudate, if not completely dissolved and absorbed, would be organized by granulation tissue, leading to fibrous adhesion between tissues. For example, pericardial adhesions. Now, let's talk about proliferation. Proliferation occurs both in the parenchyma and mesenchyme. Examples of the former include the proliferation of epithelium and glands. For the mesenchyme, we have the proliferation of macrophages, endothelial cells, and fibroblasts. Actually, the proliferation of endothelium and fibroblasts usually forms granulation tissue, resulting in fibrosis. The beneficial effect of proliferation is to repair damaged tissues. Of course, the best way is complete regeneration of parenchymal cells. But more often, repair requires the formation of granulation tissue. However, excessive proliferation can destroy tissue by causing fibrosis. Excessive proliferation could also result in the formation of inflammatory polyps. I will introduce inflammatory polyps in another session. Okay, let's summarize. Inflammation is a defensive response of living organisms with a vascular system to all forms of injuries. Inflammatory reaction may localize and eliminate the cause of cell injury, remove necrotic cells and tissues, and initiate the process of repair. The basic pathological changes of inflammation include alteration, exudation, and proliferation. Among them, exudation is the most important feature, exerting potent protective effects. However, inflammation can also be potentially harmful to the body. That's all for this class. Here are the references. Next time, we'll talk about the process of acute inflammation. See you then!